So I'd like to introduce Dr. Amal Boukteb. Amal holds a PhD in biological sciences from the National Institute of Agricultural Research in Tunisia and the University of Tunis El Manar. She earned her master's degree in genetics and a master's degree in biostatistics, and a PhD focused on population genetics and gene expression in plants. Amal has been a very proactive member of the genomics community in Tunisia and beyond, and we're incredibly impressed with how dynamic Amal has been. She's a co-organizer of the Tunis R user group and a Carpentries certified bioconductor instructor as well, dedicated to promoting open science through workshops and training in bioinformatics and data analysis with a focus on R and bioconductor. So it's a real great pleasure to host you, Amal, for this seminar, the first in our series. Thank you.
Thank you. Thank you, James. Thank you for the invitation, and thank you for this introduction. So today I will present my PhD project that I recently defended last December. So I will present how bioinformatics tools helped us to understand the interaction between Vicia faba and Orobanche foetida. So today I will talk about one of these beautiful plants. So maybe at first look, you want to have it in your garden. However, as captivating they are, these plants are parasitic, relying on a host for their nutrient, water, and resources. This plant use specialized organ called hostoria to penetrate the host tissues and establish a vascular connection, drawing everything they need from their host. There is two main types of parasitic plant. The first one is the holoparasite, like Orobanche species, lacking chlorophyll and depend entirely on their host for survival. The second type are the hemiparasite plants, such as Striga species. Despite that they contain chlorophyll and can perform photosynthesis, they still rely on the host for additional nutrient. There are about 292 genera and more than 4,000 species of parasitic plants, but fortunately, only 25 are pathogenic. So today I will talk about one of them, Orobanche foetida, this red one. It's a holoparasitic plant that completely lacks chlorophyll. This means it relies entirely on its host for all nutrient and it cannot perform photosynthesis. Orobanche foetida is a tetraploid species, which may complicate the genomic analysis. And in Tunisia, it's particularly aggressive on faba bean, Vicia faba, causing significant yield losses. And in heavy infested field, these losses can reach up to 90%, posing a serious threat to agriculture. Beyond Tunisia, Orobanche foetida is attacking wild plants, wild legumes and plants in regions like Spain, Portugal, and Morocco. The life cycle of Orobanche species is entirely dependent on its host plant. When the parasite seeds detect chemical signals released by the host, mainly strigolactone, and in case of Orobanche species, it's orbancol, they begin to germinate. After germination, the parasite form a hostorium, a specialized organ that penetrates to the host root system. This process is also guided by host-derived chemical, allowing the parasite to absorb nutrient and water from the host. During the underground phase, Orobanche foetida forms tubercles, making its detection difficult until the stem emerges above the ground. Once above the ground, the plant produces flower and new seed, which can spread and germinate in the presence of a host plant. These seeds can remain dormant in the soil up to 20 years. The complexity of the life cycle of Orobanche foetida highlights the significant challenge in controlling this parasite, especially as it poses a major threat to crops in Tunisia, and especially for faba bean, with yield losses reaching up to 90%. However, effective management of Orobanche foetida remains limited to a lack of data on the genetic structure and diversity of the Tunisian population, as well as gaps in understanding the molecular mechanism of response in Vicia faba. So to address these challenges, it's crucial to adopt a comprehensive approach combining studies on the genetic diversity of the parasite with investigation into the host response mechanism. Therefore, the objective of my PhD project were divided into main areas. The study of population structure and genetic diversity of 18 population of Orobanche foetida, collecting from faba bean infested field using INRAT technology. The second part was about exploring the gene expression of two faba bean variety, one resistant and one susceptible during Orobanche foetida infection using RNA technology. So just let's start about our first part. So understanding the genetic diversity of Orobanche foetida is crucial for crop improvement program. This knowledge helps to predict the evolutionary dynamic of the parasite population and guides to develop resistant variety. However, Orobanche foetida remains underexplored from a genetic perspective. While classical markers such as RPD and AFP have been used, no specific markers have been developed for this species. To overcome this limitation, the RAT-seq represents a powerful tool. This high-throughput sequencing method allows the generation of highly informative SNP without prior knowledge of the target genome. So our methodological approach consists of collecting 18 populations of Orobanche foetida, including 244 samples from highly infested faba bean field in northern Tunisia. Following the sample collection, we extracted DNA from fresh Orobanche foetida floral bags. Then we prepared RAD library through 3 main steps: enzymatic digestion, adapter ligation, and PCR amplification. DNA fragments were approximately 350 per base were selected for single-end sequencing using Illumina platform. The sequencing read generated were analyzed following this bioinformatic pipeline. The low-quality sequence adapters were removed using the Trimomatic program. And for the SNP calling, we employed 3 different pipelines: PIRAD, STAX, and UNIQ. For the population structure analysis, we also used several complementary methods, including test3, fast structure, GFPC, and PCI. And then we analyzed the genetic diversity, the analysis of molecular variants, and the isolation by distance effects. Some of these analyses were performed on a Unix-based system using high-performance computer, while others were conducted in RStudio on my personal laptop. So just to give you an idea about it. So as I mentioned earlier, we used 3 different pipelines for SNP calling: PIRAD, STAX, and UNIQ. These pipelines produced varying numbers of SNP as expected due to the difference in their approach and algorithm. PIRAD pipeline identify homologies regions perform well with highly divergent samples and account for indel variation. It generates 1,785 SNP. STAX is designed to distinguish true polymorphism from sequencing error and succeed to generate about 10,755 SNPs, while UNIQ is particularly efficient in managing paralog genes and resolving issues related to repetitive region, and it's well suitable for polyploid organism, and it succeeds to reveal 206 SNPs. This variation in terms of number is not unusual and aligns with pattern observed in other studies where UNIQ produced 82% more SNP compared to STAX in STAX. It's important to mention that pipelines with the reference genome usually generate more SNPs than those without a reference genome. So after with SMP identified, we turned our attention to explore the population structure using various clustering methods, the IPC, FASTA structure, and test3. And we applied this method to the three datasets, PIRAD, stacks, and unique. And to compare clustering patterns, we superposed p-chart of average membership probability on geographic map. So the main observation is that some population showed clear genetic differentiation across dataset and methods. So as you can see, the light blue cluster is dominated in population 7 using the 3 clustering method and for the 3 datasets. While the black cluster was dominated in Genduba population using test3 clustering for the all dataset and GPC and fast structure for unique dataset. Also, the green was more frequent in Beja governorate population with varying variation across the dataset and method. Also, we can see that the yellow cluster is characterized for the population from Siliena and Cave. So after this observation, we proposed to divide this population on 4 groups. So one group for population 7 from Tunis, group for populations from Beja, a group 3 for Genduba population, and group 4 including Cave population and Siliena populations. So PCA confirmed the divergence of population 7, especially with PIRAD and stacks dataset, while for the others, we observed slight diversions, but mainly overlapping regions for the Northwest population. So next, to further explore the structure of this population, we used bar plot illustrating the clustering result from test 3 for key value 4 and 6 in order to explore more additional grouping. In fact, test 3 is known for its integration of spatial data and its robust detection of structuring, even with low divergence. For PIRAD and stacks, we observed similar grouping by the distinguish of population 7, but for the others, no really strong clustering. While for unique, we couldn't distinguish any clear clustering even for population 7. So to better understand the genetic divergence among this population, we calculate the FST value, which allowed us to quantify the distance between the group. Overall, we observed low FST value across all the datasets, indicating relatively low genetic differentiation between most groups. And the lowest FST value, as expected, were between group 2 and 3 and group 2 and 4 in the Northwest region. However, the FST value confirms the divergence of population 7 by high values between group 1 and the others group. So after the evaluation of the population structure of the 18 studied population of Orobanche foetida, we focused on genetic diversity. The allelic richness was 1.2 for unique and 1.06 for stacks and PIRAD. Overall, we observed the observed heterozygosity was lower than the expected heterozygosity, indicating a deviation from Hardy-Weinberg equilibrium. Additionally, we observed a low positive value for the inbreeding coefficient. So, similar results were reported for Orobanche cumana in Turkey, Spain, and Bulgaria using SSR markers where low genetic diversity and signs of inbreeding were also observed. Next, we focused on the analysis of molecular variance that revealed that 96% of the genetic variation occurred among population, while only 3% was observed within population across the three datasets. This suggests that an autogamous behavior of Orobanche foetida. In fact, Bill and Roman studies indicated that anatogamy behavior of Tunisian population of Orobanche foetida attacking crops, whereas the Spanish population attacking wild plant showed anagamy behavior. A similar divergence was observed in Striga. However, no significant differences were detected for Orobanche chimera. To further investigate the origin of the observed genetic differentiation, we performed an isolation by distance analysis which revealed a positive correlation between the genetic and geographic distance. This pattern was consistent with all the three pipelines, pyrite, stacks, and unique. In fact, the genetic difference of population 3 appears to be linked to their geographic separation, with distance ranging from 26 to 76 kilometers from other population northwest. But also, the population 7, which is located in 185 kilometers from the others, showed the higher genetic divergence compared to the other three groups. This observation allows with finding in other parasitic plants. For example, the divergence of Striga population separated by more than 100 kilometers has been reported in Ethiopia and Nigeria. So to conclude the RAT-seq analysis, our results have provided valuable insight into the genetic diversity structure and differentiation of Orobanche foetida populations in Tunisia. This finding highlights the autogamous nature of the species, the impact of geographic isolation on genetic divergence, and the absence of strong clustering pattern across different datasets and analysis methods. Overall, this study sets a foundation for understanding the dynamic of Tunisian Orobanche foetida population. So now moving forward to the second part of my PhD project, focusing on exploring gene expression in two faba bean variety during Orobanche foetida infection. So our methodology for this study aimed to compare gene expression between two faba bean variety during Orobanche foetida attack. Given the fact that the most critical interaction occur underground, we adopted an in vitro co-culture system, which allowed us to easily track the progression of Orobanche foetida development. We used two faba bean variety, one susceptible, known as Vicia, and one resistant, known as Shuruk. Roots were collected at 3 key stages of Orobanche foetida development: before germination, after germination, and during the tubercle stage, under two conditions: in the presence of and in the absence of Orobanche foetida. Following the sample collection, we conducted a total RNA extraction, selected mRNA, and prepared cDNA library for high-throughput sequencing using Illumina platform. For the bioinformatic analysis, we adopted similar strategy to the RAT-seq project, using multiple de novo assemblers, so de novo, trans, Oasis, CNC, and Trinity. The quality of the generated assembly was evaluated using Busco, RNA-Quest, and HiSet tool. The best quality assembly was then selected for the downstream analysis, which included read mapping, clustering gene count, annotation, ortholog search, gene ontology, differential gene expression, and exploring some specific marker that show to be implicated in this kind of interaction. While for gene expression analysis and the gene ontology were performed on my laptop using ALT packages, or other analysis were performed in Linux environment on a high performance computer. So to identify the most suitable for assembly for our dataset, we tested 4 assemblers: SOAP, Trinity, Oasis, and CLC. Each assembler has strength and limitation confirming that no pipeline is universally affected across all situations. For qualitative analysis using RNA-Quest, in terms of the number of transcripts, OASIS produced the highest, while CSC generated the lowest. SOAP and Trinity generated the highest N50 value, indicating longer contig, whereas OASIS produced the shorter transcript. SOAP generated the longest transcript for the susceptible variety, while CLC performed better for the resistant variety. For the qualitative analysis using USCO, SOAP had the best result for complete genes, followed by Trinity and CLC, while OASIS failed to detect any complete gene. Finally, bit alignment results showed that Trinity achieved the highest alignment rate at 82%, followed by SOAP, CLC, and OASIS. And therefore, we will continue the following analysis using the Trinity assembly. So in our, we proceed next with the differential expression gene, and we identified 622 genes for the susceptible host and 381 the gene differential expression for the resistant host. We also observed common unigene across all the development stage only for the susceptible hosts. Additionally, based on the ortholog search analysis, we can conclude that each host has a specific set of differentially expressed gene, where genes that differentially expressed in one host were found to be insignificant in the other hosts. So to further understand the role of these differential gene expressed gene, we conducted a gene ontology analysis. In the susceptible variety Vicia, we observed an overexpression of gene ontology terms linked to secondary metabolite, particularly flavonoid, which are known to stimulate hostilium formation in root parasites. While for the resistant host Shuruk, we observed the overexpression of gene ontology terms associated with thiamine, which play a role in stress response, although its accumulation does not always translate into resistance. Additionally, actinophylamine, which are crucial for plant immunity through structural reorganization, were also overexpressed in Shuruk. This finding highlights the distinct molecular response between the two host varieties during Orobanche foetida infection. As part of the always ongoing exploration of the gene expression in response to Orobanche foetida infestation in Vicia faba, hormonal signaling emerged as a key element requiring particular attention. Indeed, jasminic acid and salicylic acid are essential for identifying the plant's response to the pathogen. Salicylic acid is employed usually against biotrophic pathogen, while jasminic acid is involved in response to nectotrophic pathogen. Our result revealed the overexpression of several jasminic acid-related terms in the resistant host. However, only one term with a non-significant p-value was detected in the susceptible host. In other pathosystems, sunflower perceived Orobanche cumana as biotrophic pathogen due to the overexpression of salicylic acid-related gene, while sorghum treated striga as nectotrophic pathogen following the interaction, the inactivation of such gene. In case of Vicia faba and Orobanche foetida, at this level, it's difficult to draw a conclusion based on the obtained result. For the resistant host, the overexpression of jasminic acid-related terms may suggest a response associated with necrotrophic stress. However, the absence of Orobanche foetida attachment complicates the interpretation, as this could reflect an effective preventive response rather than an infection-related response. For the susceptible host, the non-significant overexpression of jasminic acid terms suggests weakened response, but the lack of significant activation of salicylic acid-related terms does not point toward biotrophic recognition either. So we decide then to target more genes that already reported to be involved in parasitic plant interaction. So next, we focused on the worky genes family, and we successfully detected the expression of 30 worky genes out of 20 detected. And our result revealed that certain genes were immediately expressed in either a susceptible host or the resistant host, while some genes were commonly expressed across the two varieties. We also detected the exclusively expression of 4046 in the susceptible host, suggesting a potential role in salicylic acid signaling. Overall, when comparing this finding to the sunflower Orobanche pathosystem, distinct differences emerge, highlighting the specificity of each host parasite interaction. So in our study, the resistance of the variety shrew was characterized by reduced or complete absence of Orobanche seed germination. So, since germination depends on host-derived signal mediated by stereogalactone, and in case of Orobanche species is orobancol, we decide to investigate the expression of genes involved in orobancol biosynthesis pathway. There are two potential pathways for biosynthesis. The first one is indirect, observed in rice by converting the CLA to 4DO via CP711A2, and then converted the 4DO to orobancol by the CP711A3. The direct pathway consists on converting the CLA to orobancol via the CP722C, and this pathway was identified in cowpea and tomato. So our results showed that the susceptible host expressed the CP722C, promoting the production of the orobancol and facilitating the Orobanche infection, while the resistant host suppressed this gene and activate the CP711A3 and reduced the orobancol production and limited the infestation. This result aligns with the finding by Trabelsi, which demonstrate that the orobancol was present in the root exudate for the susceptible faba bean variety Bedia. In contrast, it was undetectable in the resistant variety. This finding highlights that orobancol is a pivotal compound in the plant's early defense strategy. And to understand the origin of this expression difference between the two varieties, we analyzed the presence of these genes in the genome of these varieties using simple PCR. And our results showed that all genes were found in both genome, indicating genomic uniformity, and that the difference lies in gene expression level. This opens the opportunity to develop resistant variety by targeting reduced germination, especially through the inactivation of the CP722 gene. In fact, similar strategies have been successfully developed in other pathosystems, including the mutation of NGCS1, which reduced the striga germination by modifying the dominant stereogalactone in sorghum root exudate. Additionally, the mutation of CDD8 in tomato reduced the reproduction of orobancol and solanacol. Recently, the inactivation of CP722C gene via CRISPR reduced the production of orobancol in root exudate of tomato and limiting the signal required for the parasite germination. This approach highlights the potential of gene inactivation by targeting susceptible gene as a tool to control parasitic infestation in crops. So overall, for this project, we studied the structure and genetic diversity of Orobanche foetida, and we found genetic homogeneity among this population, despite some differentiation driven by geographic distance, which is promising pattern for breeders to develop their breeding program. The autogamous pattern of behavior of Orobanche foetida confers an evolutionary advantage, facilitating colonization and adaptation. So the second part of this project was about the gene expression analysis in Vicia faba, and the resistance of our host was characterized by the absence or limited germination seed of Orobanche in the presence of resistant host. The gene ontology analysis and the expression of working genes revealed host-specific response for our pathosystem. And the most interesting finding in the study that the exclusively expression of CP722C in a susceptible variety offer a promoting target for the developing resistant variety by reducing parasite germination. So this project was conducted thanks to a Tunisian-Japanese collaboration between the laboratory, field crop laboratory at INRAT Institute and the plant immunity group Genshiraso lab at Riken Institute. And this collaboration was coordinated by my PhD supervisor, Dr. Amal Boukteb. I would like to thank them for their support and all the opportunity that offered to me. So just to resume how this journey went through. So we collected the samples in Tunisia and prepared for RATSEC and RNA-seq project. Then I moved to Riken, Yokohama for 3 months when I prepared the cDNA library for RNA-seq project. Once back in Tunisia, I had the opportunity to attend the Bioinformatics and Genome Analysis course at the Pasteur Institute of Tunisia. Then I went back to Japan to learn and analyze bioinformatic data analysis for RATSEC and RNA-seq project. And I had the opportunity to collaborate with scientists from Kyoto University. And thanks to the remote access to Riken HPC server, I continued to analyze my data from Tunisia. And through that, I had the opportunity also for a master's degree in biostatistics from the University of Sfax, and which I highly recommend this master's degree, especially it's online, so it make it easier. And 2 years ago, I joined the Tunis R user group, which is the Tunisian chapter of R user group. So now with my colleague Hedia, we organized several online workshops for bioinformatic analysis using R programming. So you can find all the previous workshop on our YouTube. And to learn more about our journey and what we want to do, so I invite you to read our interview with our consortium. And we have some specific success story within Tunis R user group, where we received more than 600 registration and more than 200 participation for our workshop bioinformatic analysis using Chatlace and ChatGPT. Also, we are so proud that our workshop with Dr. Andy Shen from John Innes Center just reached 10K views after just 1 year. So don't hesitate to reach us out and express your needs. So, and we can conduct the new workshop according to your needs. And recently, after receiving my carpentry certificate as an instructor for bioinformatics, we organized our first in-person workshop at INRAT as an introduction course for R programming. And we are really proud that it was the first bioconductor carpentry workshop in Africa that was conducted by 100% Tunisian team. So don't hesitate to reach us out and to connect with us. And with Hedia, we now thinking to conduct in parallel online workshops and in-person workshops. So, and thank you for your attention and for coming today.